Okay, so in Module 1 here, we need to start off this section on basic biological principles by kind of introducing what exactly is science. So science is defined as an organized way of using evidence to learn about the natural world. And that's actually a very purposeful definition. Science, by its nature, is very organized. Evidence, we use a lot of data that we collect, and the natural world. We're only dealing with things that are real. So, like I said, science deals only with the natural world. We're not dealing with things that are imaginary or pretend. And scientists work to collect and organize their information in a very careful, orderly way so that they can look for patterns and connections to explain what's happening. Now, the explanations that they propose have to be testable. We have to look at the evidence to make sure that those explanations are correct. So the goal of science is therefore to understand and investigate nature and use these explanations to make predictions. So based on our explanations that we've been able to make, we should be able to predict different outcomes in the natural world. So how do scientists accomplish this? Well, there's a process that they go to, but it really boils down to three basic things: observations, inferences, and hypotheses. So observations are facts or information that we obtain using our senses. So this can be really any of them: touch, taste, smell, sight. We tend to rely very much on our sight for different things like measurements, but those other senses do play a role. This is what we actually use as evidence. It helps us to notice things. It helps us to be the data that we collect. We can have quantitative as well as qualitative observations depending on the type that is needed. When we look at our quantitative data or quantitative observations, these are ones that involve numbers. Things like 2 milliliters, 4.2 inches, and 75 pencils. Qualitative data is a little harder to define because it's anything that we can't measure. And this would be things that are subjective like color, size, shape, and even texture. But both of these play a role in our use of science or our understanding of science. Sometimes a color change is important. It's not a measurement that changes. So we have to pay attention to both types of data. What you have to understand, though, is observations by themselves have very little meaning. If I say blue, it really doesn't mean anything. You have to understand the context. But observations are generally followed by inferences. Inferences are where we actually interpret the observations based on our prior knowledge and experience. So it interprets the evidence that we collect using our observations. And we use this to draw conclusions about things. Now this is frustrating because sometimes you're right and sometimes you're not, and you don't know. If I gave you a set of observations, yellow, spherical, warm, I could be talking about the sun, or I could be talking about like an egg yolk or something. There's not enough contextual clues there for you to understand. So that leads us then to the hypothesis. Hypothesis is a possible explanation for a set of observations or a possible answer to a scientific question. This is usually what we're testing in an experiment. It's trying to explain the evidence. In other words, it's a guess in terms of, okay, we're noticing this and this is why. So we can describe this as an educated guess. We're not going to make wild imaginative guesses with this, but they do have a place in science. So a hypothesis can arise from prior knowledge of the subject, a logical inference, and even imaginative guesses. They do play a role, but we're going to avoid them in this class. A hypothesis is useful only if it can be tested, and you probably want to mark that. That's a really important point about hypotheses, is if you can't test them, they're not useful and we shouldn't be using them. So we take all of this information and we put it together in a way that all scientists follow, and that is the scientific method. Now, the scientific method is basically that organized process that we use to try to answer questions. There is a series of typical steps that allows us to replicate or repeat our experiment or even combine it with the work of others. What this allows us to do as scientists is collaborate. We can work together with other scientists in different parts of the world. So we're going to quickly here go through the steps of the scientific method as a review. So our first step is stating the problem. When we state the problem, we use observations to identify and ask a question about something that we don't understand or we want to explore. So we notice something and we have a question about it. That's our stating the problem. We form a hypothesis. This is a testable solution to the problem or question from step one. It's usually in the form of an if-then statement. Sometimes you add the because if you're trying to hypothesize the reason behind your observations. The general format. If part of the statement contains what we call the independent variable. That's the one that I, as a scientist, control. The then part of the statement shows the dependent variable. That's the one that I can't control, and usually I'm trying to figure out if that's right, if that's what's changing, if it's changing in the way that I think it should. Because tries to explain the evidence, so if it's known. So an example of a hypothesis would be if I lower the temperature of the freezer, then the water will freeze faster. I can do that. I can control the temperature of the freezer. I can change it. And I'm hypothesizing that if I turn it down, the water will freeze faster. Now in this one I don't have a because. I could throw out another guess, but right now I'm just guessing about what's going to happen to that dependent variable. How fast is the water going to freeze? I think it's going to freeze faster. Step 3, we set up a controlled experiment to test the hypothesis. When we set up our controlled experiment, first we need to make sure that we have all the materials that we need for this experiment. We create a list of numbered steps, which we call the procedure. So this is exactly what you're going to be doing during that experiment. You will also have at least 2 setups in most of our experiments. You're going to be testing 2 different groups of setups, or two different groups of or situations of variables. The first setup is called the experimental group. Now in this group we actually will change the independent variable from normal. So this would be the temperature, or the freezer with the temperature that's been lowered. Okay. The second setup is what we call the control group. This is the group that's under the normal conditions. And we need this because without this comparison, we can't tell if the experimental group is actually doing what we guessed it would in our hypothesis. So for example, the freezer set at the factory set temperature would be our control group in this situation. Now, other than that independent variable, in our example that we've been working with, it's the freezer and the water, everything else needs to be kept the same or controlled. So the size of the freezer would have to be the same, the type of ice cube tray, and the amount of water we put in each well of the tray. All of those would have to be the exact same in order for this experiment to really show that it's the temperature causing the difference, not anything else that we might be changing. If we change too many variables, if we change more than one variable, then we can't really tell what's causing it. Now, we have to include safety precautions as part of it. Safety is always first and foremost. And if there's no specific safety information, then we just need to recommend some general safety concerns that might be with that particular experiment, like wearing goggles, wearing closed-toed shoes, those types of things. In your controlled experiment, you need to identify what data you want collected. Are you going to collect how much time it takes, or how many ice cubes it freezes in a certain amount of time, or what kind of data are we going to be collecting? So you need to include how it should be collected. Are we going to be taking a temperature? Are we using a timer? And when it should be collected? We're going to do it every 5 minutes. We're going to, how is this going to be done? So we could do, for example, time in minutes until the water is slushy, and then the time in minutes until it is frozen solid. In the experiment, we also need to make sure that we try to include a way that, maybe next time we could improve the reliability or validity of the data. Couple of ways that always work: repeat the experiment. That's a really easy way. You could also do more trials. That would be having more trays of ice cubes, because maybe that one particular one is a little different. You could also test at other temperatures, so you could have more setups. So maybe I have one that's at -20, one that's at -40, and so on. One at -10. See which one actually freezes water the fastest. Can it be too cold? So as we're collecting this data, we need to make sure that we are recording it. So we need to record and analyze our data. Now data, a lot of times we abuse, but data should be recorded in a data table. We need to have a nice, neat way of, organized way of recording this data. Now when we analyze it, we look at the patterns and connections, and a lot of times this includes making graphs. Graphs are important. They are a pictorial representation of our data, and it's really important that we know how to make good graphs and we know how to interpret graphs. Once we have run the experiment, we've recorded our data, we have analyzed our data, we can draw our conclusion. This is where we actually point out the patterns in our data. We identify whether or not our hypothesis was supported, but we don't use the word prove. We're not proving anything with experiments that we do in here. We're only supporting or rejecting our hypothesis. The reason why is in order to prove something in science, you have to run the experiment many, many times. Running it one time doesn't cut it. So don't use the word prove in your answers. You're going to identify sources of error, and these aren't mistakes that you make per se, but they are built-in things in the experiment that don't allow it to maybe run the proper way. You can propose additional experimentation to help solve the problem if maybe you get an answer that's inconclusive, or you can answer new ones that have developed. Lastly, you want to make sure you report your findings. If you don't share your information, then it doesn't help anybody. I often use the example, if I come up with a cure for cancer, that's wonderful and that's great, but it's never going to benefit the people who need it the most if I don't share it. It's also our way in science of keeping each other honest. So other scientists will check out my work and make sure that, yes, she really did come up with a cure for cancer. Now, that is how we run an experiment, but we also have some things here in science that are more than just a single experiment. And this is where we come into the ideas of scientific theories and scientific laws. A scientific theory is a well-tested explanation for a set of observations or experimental results. This is not just a guess. Remember, in science, we use the word hypothesis for our guess. Out in the real world, we often talk about things being just a theory because we think that they're just a guess, but we don't do that in science. This is based on repeated tests. Theories can be revised, and that happens all the time. And it explains why something is happening. So, why does evolution happen? Why does the atom behave the way it does? We have the atomic theory and we have the theory of evolution, both of which explain that. The second one is a scientific law. Now, sometimes people think laws are better than theories. It's not that they're better, they do different things. This law summarizes the pattern, so it tells what's happening, and it's a pattern that seems to be true all of the time. In other words, we haven't found any exceptions to that particular law, like Newton's laws of motion. It is talking about what happens. So it's not explaining it. I know it says the word explains, but it talks about what happens. For every action, there's an equal and opposite reaction. It doesn't explain why. It's not a theory. It's a law. It is the pattern. Every time we have an action, we have an equal but opposite reaction. And that's what a law is. So this was a very brief review of your scientific method and kind of a reintroduction to how we work in science.